In this video, we will be explaining the process of meiosis. Meiosis is the production of gametes from one diploid cell, occurring in 2 stages, resulting in 4 genetically different daughter cells. Here are some definitions that will help with your understanding of the video. A gamete is a male or female reproductive cell. This can be compared to somatic cells, which are every other kind of body cell. A diploid is a cell that has 2 sets of chromosomes. A haploid is a cell that has 1 set of chromosomes. A chromosome is DNA that's tightly coiled around a protein called a histone. A chromatid is 1 copy of a newly copied chromosome. What we see here is interphase, the phase right before the start of meiosis. The cell is preparing for reproduction. Within interphase, the G1 phase creates proteins for cell growth. The S phase duplicates the cell's chromosomes to create identical sister chromatids. The first stage of meiosis begins with prophase 1. The nuclear membrane dissolves. The chromosomes begin to condense and form bivalent or homologous pairs. This is known as synapsis. The homologous pairs exchange parts. This is called crossing over. Crossing over will cause the daughter cells to become genetically distinct. Next is metaphase 1. Spindle fibers form and attach to one of the chromosomes in each pair, lining up in the middle of the cell in random orientation. In anaphase 1, the spindle fibers pull to opposite poles with the homologous chromosomes moving as well. The sister chromatids stay attached. With telophase 1, the chromosomes arrive at the opposite poles of the cell. The cell divides, which is called cytokinesis, forming 2 haploid daughter cells. The second stage of meiosis begins with prophase 2. The chromosomes begin to condense and form homologous pairs, creating chiasma or an X shape. The chromosomes move to the opposite sides of the cell and the spindle fibers form. In metaphase 2, the chromosomes line up individually in the equator or middle of the cell. In anaphase 2, results the sister chromatid separating and moving to the opposite sides of the cell. During telophase 2, the nucleus reforms and the chromosomes decondense. Cytokinesis divides the cell, forming 4 genetically different haploid daughter cells, and meiosis is finished.